Okay. I still see some are joining, but I think we can get started slowly by introducing this event. So I'm Remi Dangla. I am the CTO and founder of Stilla Technologies, and today I will be chairing this event as part of ASHG Industry Forums, an event called Advancing High-Plex Digital PCR into the Mainstream. I am very pleased to have three guest attendees with us today. Each of them will give a presentation on their research topics. So we have Herman Cornelius from the University of Washington. We have Evie Lianidou from the University of Athens. And we have Lao Sal from the University of Lund, and also a founder of SAGA Diagnostics in Sweden. We will have this event made as much as possible interactive. We will have the speakers present each one after the other in the first part of the presentation, and then finish with a Q&A session at the end to discuss in general the topic of high-plex genetic analysis and the context of research activities and clinical applications. Before we go into our invited speaker talks, I will give a brief introduction to the topic of digital PCR and introduce also why we are discussing this topic of high-plex digital PCR. So let me share my screen, and I will ask one of the guest speakers to confirm that the screen is showing.
Yes.
Okay, thank you very much. All right, so as I said, today we are all gathered to discuss a very exciting and interesting topics, the topic of advancing high-plex digital PCR in the field of molecular diagnostics. And before we go into the details of high-plex digital PCR, I just want to make sure that everyone in the audience is well aware what digital PCR is about. Digital PCR is the third generation of PCR following endpoint PCR invented in the 90s, Qpcr in the early 2000s. Digital PCR really brings some advantage over competing PCR technologies such as sensitivity and precision in the analysis. The way digital PCR works is actually quite simple in its principle. So when you do digital PCR, you start by taking your bulk sample, which contains all the DNA that you have to analyze. But instead of doing the analysis in bulk, you start by splitting the sample into tens of thousands of partitions. And as you split the sample into those partitions, you're basically separating the DNA molecules in different compartments. So you start out with a complex mixed sample, and you end up with partitions that are each contain one or very few DNA molecules, which makes then the PCR amplifications much cleaner and much more robust. So once you've done the partitioning, you amplify those partitions by PCR, and you read out the content of those partitions by fluorescence. So the partitions will either be non-fluorescent, they will be empty, or fluorescent, meaning they contain one or more DNA of interest. You then detect the fluorescence on those partitions, and you count the number of positive and negative droplets in your sample. Applying some Poisson statistics, you can then easily extract the quantity of DNA molecules in your sample, and you can quantify those DNA molecules with a very precise number. So your precision is usually much below 10%. And you also have a great sensitivity because you can detect a single positive partition, and that will mean that you have detected a single DNA molecule. So you essentially have single molecule sensitivity, and you have near single molecule precision in genetic analysis. Digital PCR is also very robust against inhibitors and PCR chemistry in general. Those key features of digital PCR have really made digital PCR become one of the next standards in the field of molecular biology. It has expanded greatly in its use in the research labs in the past 5 to 10 years. And if you look at publications in 2021, you had more than 4,000 publications that use digital PCR as part of the work presented. And it really is being used in all fields of molecular biology, the main driving fields being oncology, infectious disease, but you also have applications for environmental testing or food testing. And you can use the technology for various types of assays, such as absolute quantifications, copy number variations, rare event detection, or gene expression. Today at Stilla, we are very excited to be hosting this event because we are launching a new system on the market called the naica Prism 6 instrument. It is this system here which really expands the power of digital PCR by taking digital PCR from a low-plex approach to a high-plex approach, meaning that we can now detect a high number of different types of DNA or RNA in a sample, and that opens up the field of applications addressable by digital PCR. This system is available today, and we will discuss today with our speakers, Evie, Herman, and Lau, how digital PCR can be used in high-plex fashions to target different applications in research and clinics. So I will first start with a quick questions to our guest speakers today and ask them, in their opinion, what is prompting the demand for multiplexing in genomics across research areas, and whether they see a link with the need for modern genomics to be deployable in the clinic. So maybe I can ask first Lau to give his point of view on this question.
Yeah, thank you very much, Remi. It's a pleasure to be here. I think that's a really good question. So I represent SAGA Diagnostics. We're a cancer genomics company focused on the cancer liquid biopsy. And we've been working with digital PCR on the research side for almost 10 years, and now on the commercial side in SAGA for about 5. And what SAGA is really trying to do is to democratize cancer liquid biopsy testing and have it widely used in the clinics. And we see digital PCR as being a major part of that democratization. Of course, NGS has a place, but for a lot of applications in oncology, it's overkill. It's too much, it's expensive, it's slow, it has a lot of steps in the lab. A lot of hospital labs don't have the sample flow and scale to make it affordable. And that's where we think digital PCR fits in. So we get a lot of interest from the market in digital PCR and what we have to offer, especially for clinical applications in oncology. And we're working with a number of global pharma companies and hospitals to make that happen. So single genes and 1 or 2, mutations is sort of where things started out a bunch of years ago. But of course, there's a need for having more targets in your test. NGS that can do hundreds, like I said, is probably overkill for many clinical applications, but having tests that can do, a dozen or several dozen, I think there's a real sweet spot for that. With digital PCR in the clinics.
Thanks, Lau. Herman, do you want to share your point of view?
Sure. So I think, in our lab, I represent the University of Washington, and I work for the Department of Obstetrics and Gynecology. So I think, essentially, we can use dPCR for 2 purposes. One is in terms of sexually transmitted diseases like HIV/AIDS. We use dPCR to quantify the size of the HIV reservoir in people. So it's a high throughput way that we can test different curative approaches for HIV. We are using different anti-proliferative methodologies that are cutting edge. So we can easily and quickly measure the HIV reservoir. But it's more importantly, we can also characterize the structure. So we can see in a single droplet different genes of HIV, and then we can see if the HIV provirus is intact or not. This is the case of HIV. We are also developing a new chlamydia assay to test samples, which is also very quickly in the clinic. In terms of opioids, we are also trying to understand how different opioid therapies, to treat opioid addiction, how they change the gene expression. So it could be another way to measure very quickly. And lastly, we are using also Crystal DPCR to see how different medications to treat HIV that are known as antiretroviral therapy or ART, how they also activate the immune system. So we can multiplex different genes of interest.
Okay, fantastic. Evie, I'm sure you're going to touch that in your presentation, so maybe for the sake of time, I can turn it over to you, and I think you'll address that point directly in your presentation. So Evie will talk today about a very interesting study where she looked at EGFR mutations both in cell-free DNA and CTCs in paired samples. So I think that's really exciting, and turning it over, Evie, for the presentation.
Thank you very much. Do you see my screen?
Yes, perfect.
Okay, perfect. So my talk today is on the potential of digital PCR and Crystal Digital PCR specifically in the liquid biopsy field. And liquid biopsy is coming to give another approach in respect to classical biopsy, and it's based mainly on the analysis of circulating tumor cells and circulating tumor DNA. There is a lot of information that we can get from liquid biopsy through serial blood analysis for various markers. And we can get from liquid biopsy information from peripheral blood on CTC at the protein expression, gene expression, and DNA level, epigenetic alteration as well, ctDNA also in mutations, amplification, point mutation. And from cell-free DNA, we know that it's a sub compartment of circulating tumor DNA is a subcompartment of cell-free DNA, but we can get a lot of information. But the amount of sample that we have there is very limited. And we also have to get through very low detection limits. And based on that, to detect mutations for a number of genes or fusions of or methylation markers, we do need high sensitivity. So I'm going to talk especially for non-small cell lung cancer as an example. And I have to say that liquid biopsy comes here for treatment selection, for early detection of resistance and monitoring disease burden. We already have clinical applications, but it can also be used for early diagnosis and minimal residual disease. There are also some FDA-cleared tests on ctDNA analysis in liquid biopsies, mainly based on NGS and real-time PCR for specific genes and ZAN companion diagnostics for specific drugs. But in any case, liquid biopsy is a very useful tool in non-small cell lung cancer. And Crystal Digital PCR, here you can see Nature cover some years ago, and the FDA approved test for EGFR mutations. In our lab, we are doing a lot of work in liquid biopsy at the gene expression level for CTCs, microRNAs, DNA methylation, and DNA mutations. And we are mainly using RT-Qpcr. We're developing assays and RT droplet digital PCR, and as well a Crystal Digital PCR for mutation detection. We have ISO for EGFR mutations in cobas, in Roche, the FDA-cleared assay, and for CTC enumeration using the cell search system. Now, using Crystal Digital PCR, we have recently shown that we can detect EGFR mutations in plasma cell-free DNA and paired CTC of non-small cell lung cancer patients before and after osimertinib therapy. We used Crystal Digital PCR as a tool to evaluate the mutations of more than 42 mutations in EGFR gene. There are a lot of publications showing that EGFR mutations are highly important in non-small cell lung cancer. There is a specific drug, osimertinib, that is now given to patients that have T79 mutation. They are positive for T79. And this is now being evaluated, and novel information is collected concerning the resistance mechanism to osimertinib treatment. And based on that, we started doing a clinical, based on a clinical trial, we collect sample at baseline every 6 months and at progression of disease. And from this blood, we are doing gene expression analysis for circulating tumor cells. We have already published this, and it has to do with EMT and PD-L1 markers that have a dynamic role in these patients under treatment. And then we moved on to mutations detection using the Crystal Digital PCR from naica. So that's what I'm going to present to you, this application of Crystal Digital PCR in liquid biopsies, both in CTCs and circulating tumor DNA. We have CTCs that were isolated using a size-based microfluidic device, the Parsortrix, and we have also 48 patients with EGFR mutated lung adenocarcinomas that were treated with osimertinib. And as you can see here, using the same blood draw, we isolated plasma, where we did the circulating tumor DNA analysis for EGFR mutations, and we also isolated circulating tumor cells from the same blood draw. In plasma, we did EGFR mutation analysis using kits from ID Solutions and using the Crystal Digital PCR system for EGFR mutations. And at the same time, we compared in the same plasma samples for 80 of these patients using the FDA-cleared Qpcr assay that we have an ISO 15189 certification in my lab. At the same time, we have used the Parsortrix system to isolate CTC, and then we did also see EGFR mutations in the CTCs. I will skip that for the sake of time, but we have used the kits from ID Solution for extraction of cell-free DNA and quantification and for EGFR analysis. And this is the Crystal Digital naica system from Stilla, where we use this system, which was, as Remi said before, capable of using a number of droplets and doing single digital analysis. And by doing this and using the Crystal Miner software, we could actually detect EGFR mutations in our sample. An important issue in this analysis is the use of a positive control of extraction that we followed over different runs and over different days. And you can see here how good is the performance of the whole analysis, because these Levy-Jennings graphs can show you that the evaluation of the extraction process and the normalization are highly reproducible. So the internal control is used to standardize interassay fluctuations. And then by doing this, we have shown that the cell-free DNA as copies per microliter are higher in patients with non-small cell lung cancer, both at the baseline and at progression of disease. And here you can see some results on cell lines derived genomic DNAs for L858R mutation for EGFR. Wild type is here. This is just to control the whole system. But here you can see in cell-free DNA samples where you can see that the number of positive droplets are much less than in the cell line, of course. Here's the wild type, and here is a specific mutation. And this is before and after whole genome amplification that we also did. This is the T790 mutation, how we detect this by using droplet digital PCR. And again, S768I mutation in CTC-derived genomic DNA. So we have tested the system. We have seen that we could easily detect the mutations. Here are some examples of how we see the crystals. These are EGFR wild-type blue crystals, and here you see deletion 19, which are the green crystals that are not easy to detect, to see here, but we can actually see them in the screen here you can see C79S, the red crystals, 1, 2, 3, 4, 5. So by doing this, these are the blue crystals, wild-type EGFR. We were actually in the position to detect specific cell-free DNA fluctuations at baseline and at progression of disease. You see in all samples, progression of disease, we had high values of cell-free DNA circulating in terms of baseline. And most importantly, in individual samples, we have seen that at progression of disease, specific mutations arise at much higher percentages than at the baseline. These are for all individual samples. We have some samples like number 7, where the mutation can be detected only in progression of disease, or some other samples like L85R, number 3, number 30, number 32, we can see mutations only at progression of disease. This is a highly important information in a quantitative format, what's going on. And the percentage of mutation allele frequencies is highly important and indicative of progression-free survival for these patients. Moreover, when we detect it and we compare the system, the Crystal Digital PCR, with the FDA-cleared Roche system, we have seen a very high concordance, more than 90% in all cases for each mutation tested. Moreover, we have seen a number of cases where, like here, where EGFR T790 mutation was detected only by droplet digital PCR and not by the FDA-cleared assay from Roche. Moreover, here you can see what's going on in the primary tumor, in the plasma cell-free DNA at the baseline, in the plasma cell-free DNA at progression of disease, and in the corresponding CTC-derived genomic DNA at baseline and progression of disease. There are some cases like patient 10 where we see exactly the same profile in all compartments, but other cases where we detect mutations only in the CTCs, like here, which is a novel mutation, L858, was detected only in CTCs, while in other cases we detect mutations, novel mutations in plasma cell-free DNA. So this is a complementary analysis where we can actually see the potential and the multiplexing of Crystal Digital PCR in liquid biopsies. A comparison with what is seen in the primary tissue and ctDNA gave about 70% concordance in most cases, in some cases more. And this is also important. And the clinical significance of this is going to be evaluated after we analyze the whole number of clinical trial samples. As a conclusion, I would like to say that Crystal Digital PCR allowed us to track tumor evolution through the detection of low abundance mutations in cell-free DNA and CTCs that are predicted for the treatment outcomes of non-small cell lung cancer patients under osimertinib treatment, that this technology exhibited high concordance rates in correlation with the FDA-cleared cobas technology. However, in some cases, this technology, Crystal Digital PCR, was more sensitive than other methods in detecting the T790 mutation, which is the key resistant mutation found during treatment with first-line and second-line EGFR TKI technologies. So the presence of EGFR mutations in paired CTC-derived genomic DNA revealed discrepancies between CTCs and tumor or cell-free DNA genotyping. Future perspectives, I strongly believe that it's now time to get the information derived from liquid biopsy analysis to be translated for patient care. And this information provided by ctDNA and CTC should be used for new theranostic applications. And for this reason, we have to use very well certified and validated analytical methodologies. So liquid biopsy analysis can give us information on minimal residual disease, on relapse, and Crystal Digital PCR is, with no question, one of the major technologies that can be used and has broad application in this area. International Liquid Biopsy Association is a highly important alliance nowadays. It brings together a lot of organizations that works toward the standardization of liquid biopsy assays to bring them into the clinic. Finally, I would like to thank my group in the University of Athens, and especially Aliki Dzifa, who is the first author in the publication that I have just presented to you. A lot of thanks to our collaborators all over the world and our funding, and acknowledgements also to Stilla and ID Solutions for helping us to set up this technology in our lab. And finally, I would like to say that we had the chance a few days ago to have the 5th ACTC meeting on liquid biopsy. That was a live in-person meeting, and 210 participants came together from 23 countries, and we discussed a lot of aspects of liquid biopsy. Thank you, and I will be ready to answer any questions.
Great presentation. I have maybe just one question for sake of time, and we maybe can answer more after the other presentations. It was interesting to see that you had different results for T790M mutations between cell-free DNA and CTCs. What conclusion do you draw from this? Do you think in the future we will need both to analyze cell-free DNA and CTCs in the clinics? And is that an easy thing to do? Is the sample prep easy to do in terms of blood handling?
We have shown in many different publications, not only this one, that CTC and ctDNA are giving complementary information, especially when we have very low number of copies of circulating tumor DNA or circulating tumor cells. And moreover, in some cases, this information is complementary. I strongly believe that it's about technology. If we see clinical utility, a very solid clinical utility, that this should be both done, it can be done. The main limitation is the technologies that we need, that should be very well validated, very reliable, and capable of analyzing low number of sample volumes and sample amounts. And based on that, I strongly believe that multiplexing and using a very sensitive technology like digital PCR will allow us to do this. And moreover, I have to say that in contrast to NGS or MPS, massive parallel sequencing technologies, PCR-based technologies and like digital PCR are more prone to standardization, and standardization is highly important to use quality controls to check the extraction and all the steps. So by limiting the number of targets that you want to test, you don't need to check all the genome. You need to check specific alterations like this example. And based on that, it will be very important to use complementary information on CTCs and ctDNA.
Okay. That's excellent. Thank you, Evie. We will now turn to our second guest speaker, so Herman Cornelius, who will present his work on not an unconventional yet very important topic, which is the look of the impact of drug abuse on semen and transfer RNAs in semen. So I leave it over to you, Herman, for the presentation.
Yeah. Well, thank you. First of all, thank you for inviting me to give this talk. I'm going to show you some preliminary data that we obtained using the Crystal Digital PCR. We are very excited. So I would like to thank the whole team, especially my director, Florian, and also NIH. This work was funded by NIH grant given by the National Institute of Drug Addiction. So the goal of the project was characterize the sperm tRNA fragments. That is a new class of small RNAs. They have been discovered like less than 10 years ago, so they're relatively new, unlike microRNAs and siRNAs that were discovered a long time ago. And the hypothesis is the fragmentation pattern and also the concentration of a specific small sperm tRNA fragments is different in men who use opioids compared to men who don't use opioids. So why we believe that our work is important? So as you know, especially in America, there is a huge opioid crisis. So the question that we want to answer is, by doing this work, is that if can therapeutic use of prescribed opioid pen medications can also result in epigenetic changes in sperm, and this change or impact later generations. Also, if these mutations, because they are epigenetic, we call it epimutations. So are these mutations reversible? And when the drug is no longer used, can they reverse back to the basal state? And what is the time frame for that reversion? And then other question is that we know if some of these men are placed on opioid substitution therapy. Essentially, they are given even methadone or buprenorphine. So we want to answer the question is which of these therapies are better, and if these substitution therapies can reverse the epimutations induced by the consumption of illicit opioids like heroin. So just in a nutshell, I want to show you the biological importance of this. So we know that sperm cells express opioid receptors. We know they express different classical receptors at mRNA and protein levels. Also, if you treat sperm with opioids, they change the motility, so they migrate lower, for instance. And some preliminary studies done in animals, especially in mouse, we know that the chronic exposure to opioids and other toxicants can change the epigenetic information, specifically in the sperm. But what is important for this talk, I'm going to talk about transfer tRNA fragments. As you know from classic or the biology, you know that tRNAs play a role in translation. But what is not known is these tRNAs can be cleaved into a shorter biologically active regulatory RNAs that we're going to call from now on TRF. The generation of TRF is highly regulated. There are many enzymes. It has been shown that they are induced by stress. So everything that has to do with like viral infections, cancer, metabolic disorders like diabetes or obesity, and exposure to insecticides or other toxicants can change the pattern in TRS. But most part of the studies have been done in mice and not in human. So how do they work? They can impair protein translation competing with the full length tRNAs. So they can block for instance, the elongation of the amino acid proteins when they're being made during translation, but also they can operate as regulatory microRNAs. And more importantly, they can also work in the chromatin level, repressing the expression of retroelements, both in germlines and embryos. But all the work, like I said, was done mostly in animals. So all these preliminary data suggests that these TRFs can potentially operate as a new epigenetic player and play a role in epigenetic inheritance. But nobody has shown the impact of opioid use in the formation of DTRS in sperm, that are germlines, in people who use drugs. In the whole presentation, this have been abbreviated like PWID, is people who inject drugs. This is just a snapshot about what we did. So we collected samples in Seattle. We collected human samples from men who use opioids. We purify and sperm cells, so we compare different purification systems. Also, we compare different RNA isolation kits. We isolated RNA from vesicles, extracellular vesicles that are called exosomes, and also from mature sperm cells. And then from that, we either created libraries to get sequenced by next generation sequencing, or we created cDNA that then we tested by Crystal droplet PCR. We use a specific method called phosphoRNA-seq that it allows us to sequence fragments that are like very small size. And we validated the libraries with Crystal Digital PCR in the lab. This is just one slide showing you how we compare different methods of isolation of sperm. The whole idea is when you have sperm cells also in the clinical samples, they get contaminated with epithelial cells that come mostly from the prostate and the epididymis. So, and this can somehow mask the true effect of opioid in sperm cells that are the ones that play a role in epigenetic inheritance. So we have to compare different methods to purify the sperm cells from other contaminated epithelial cells. That was in the beginning of our project. So in this slide, it's just a QC or quality control slide. Initially, I'm showing you an hematoxylin eosin staining, showing that our purification work. This is not purified sample. This is purified through this Percol gradient. And you can see nicely that the preparation doesn't have epithelial cells. This is also a quality control that we run is electrophoresis that shows you that in mature sperm cells, you can see a nice range of mRNA fragments. We were surprised also to find very long RNAs. You can see here, they go all the way to over 2,000 kilobases. That's actually under debate in literature. What is surprising is people who use opioids, they have lower yield of RNA, and this is already known because opioid consumption results in hypogonadism and lower quality of sperm. And here by Qpcr, we run 3 assays. One is a gene that is expressed in mature sperm cells. And you can see here the impurified sperm was enriched in this marker, which allow us to be just to confirm that our purification method work. Also, the mature sperm that is in the middle part of these graphs, you can see that the lack CD45 marker is a marker of leukocytes. So we could purify them from contaminated leukocytes. And this is another marker that is expressed in prostate cells. And also we can see that there was a decrease in that, suggesting that also we could get rid of some of the prostate contaminated cells in the preparation. This is the most exciting slide that we obtained. So we subjected these libraries. So here you're going to see different reads, and these are the alignment with the tRNAs, specifically tRNA glycine GCC 1.1. And here what I'm showing you is the sequence from 5' to 3'. And this is the abundance in heroin users versus non-heroin users. And as you can clearly see, most part of the heroin users samples were enriched in these long fragments. So this is just a summary of our slide. And what we saw is basically we saw like short fragments that the 3' end finish in the anticodon loop, and some long fragments that finish in this loop that is representative psi C loop. Okay. So in terms of the short fragment, we saw that in controls that do not use opioids, 90% of the fragments finish here in anticodon loop, whereas almost half, 45.5% of the opioid users samples finish over here. But surprisingly, we can see that most part of the opioid users, their samples, they're enriched in these long fragments, and very small percent of controls have these long fragments, and something very new, not reported in literature in humans. So what we did is we use our naica Crystal Digital PCR to confirm these findings, and we developed different Qpcr assays first, one to test for the long fragment and one to test for the short fragment. And then we tested them both by real-time PCR and by Crystal Digital Droplet PCR. And what is represented here is the abundance of the long fragments in controls and in people who use drugs. As you can see, the absolute number, this is represented in copies per microliter of cDNA. It was lower in opioid users, both the long fragments as well as the short fragments. But when each donor we normalize it, you can see that in people who use opioids, there was enrichment of these long fragments, unlike what we can see in the non-user controls. So even though they are expressing lower, both long and short, there is a higher ratio of the long fragments in people who use opioids. And then what we did is we compared and asked, for instance, we took one sample, in this case, this sample in which the ratio long-short was almost 3, and we compared, this is obtained by Crystal Droplet PCR, and then we went back to the next generation sequencing, and you can see in this donor that is extreme of the phenotype, if you will, that most part of the reads corresponds to the long fragment. So it was a total robust correlation between next generation sequencing and Crystal PCR. And also not shown here, we also ran real-time PCR, and also we observe very robust correlation between the CT values obtained by real-time and Crystal Droplet PCR. This is the last slide showing the data set that we compared. We did also next generation sequencing in seminal exosomes. The exosomes are these extracellular vesicles that are found in different biological fluids, including semen. And these are also some free-floating non-coding RNAs that are in the plasma, in the semen plasma. And then what you can see here is we ran two different runs for each marker. In light blue is the opioid users, and in red is the control. And you can see that we found a handful of microRNAs and small RNAs like these two. These are small nuclear RNAs that are expressed to lower extent in opioid users compared to controls. And thanks to the preliminary data that we obtained with Crystal Droplet PCR, I applied in 2020 in October for a grant, and this is just one email showing that the grant got awarded. And then we also apply for a grant given by the Center of AIDS Research called CFAR, and we got awarded 2, so we could purchase a 6-color naica system. And what I'm planning to do is measure different TRFs. For instance, we know in human there are 4, in addition to tRNA glycine, there are 4 more, like glutamine, valine, and methionine and lysine. So I'm planning to measure both the concentration and cleavage pattern of these TRFs in other samples. We have extra 13 more samples of men who use opioids. So I'm planning to use Crystal Droplet PCR to quantify this. And with this is my summary slide. We found in semen sample from people who use opioids, we observe an altered cleavage pattern specific of this variant of tRNA. It's called glycine GCC. And we saw that in people who use opioids compared to non-user controls. We also saw like the handful of differentially expressed small nuclear RNAs. We saw both in semen and plasma and exosomes, and we believe that this imbalance in the ratio of tRNA isoforms, especially these small nuclear RNAs, can lead to dysregulation of the epigenome, and somehow this can affect the transcriptome in semen from people who use opioids, and this can have an impact in epigenetic inheritance. And that's my last slide. Thank you, and I take questions.
Thanks a lot, Herman. That was really interesting, and I'm looking forward to seeing the data that you will get on more tRNAs. I have maybe one question for you, because it's really interesting results that you're getting, and maybe ask the question whether or not those alterations in tRNAs will continue or will reverse if someone stops drug abuse. Do you plan on looking at that as well?
It's an excellent question. Actually, I do have samples before and after Suboxone. That is one of the opioid substitution therapies. So I have longitudinal samples, and I'm definitely going to look at that.
That would be really exciting as well. All right, thank you very much, Herman. So we will turn to our last speaker today, so Lau. And Lau today will present results of a collaboration that we did with his team at SAGA and Stilla, where we tried to combine and successfully combine the high precision and multiplexing power of the naica system with the high sensitivity SAGA safe chemistry, and applied that to the detection of cancer biomarkers. So Lau, I turn it over to you for your presentation.
Yeah, thanks again, Remi. It's a pleasure to be here, and it's been a really great collaboration. So I'm going to talk about our work developing a high-plex PIK3CA mutation detection assay using our SAGA safe technology and applying it to the Stilla Prism 6-color system. Before I jump into our results, I wanted to tell you a little bit about SAGA. We are adjacent to the Lund University Cancer Center here in southern part of Sweden. We were founded about 5 years ago, and over the years, through various funding, have grown now to 17 people. We very much are focused on the cancer liquid biopsy. I think Evie gave a very good introduction to this. It's the basic idea that through a liquid biopsy sample, like a blood sample, one can measure genetic properties and biological properties of the cancer by measuring, for example, circulating tumor DNA. As a company, we are focused on the cancer patient, so we're not looking at detecting cancer in an otherwise healthy person. We're focused on the cancer patient. Once they're diagnosed, can we apply our technologies to better stratify them, to follow the patient as they receive therapy to see if the therapy is working, and detect residual disease and relapses early, and to help to steer therapy and achieve better outcomes. We have got a great team, very diverse backgrounds, of course, tons of experience in digital PCR. A lot of what I'm going to show today with the Stilla data was performed by one of our senior R&D people, Miguel Alcaide. right here. This is, well, my laser pointer, there we go. This is Miguel here. So as a company, we are focused on the circulating tumor DNA, and we have 3 different technologies to measure this ctDNA. In general, all of them are ultra sensitive. That's sort of what SAGA is known for, having super high performance methods that can measure down to the 0.001% allele fraction range. Of course, these are proprietary and patented. Because we're using digital PCR as one of the major engines of our method, this is very rapid and cost effective. It can be applied across all cancer types, and we're offering both testing services as well as research use and CE marked kits. So with the 3 different technologies, we really are covering most of oncology. What's really not the topic of this talk, so I won't spend much time on it, but SAGA Sign is a hybrid method that uses NGS and digital PCR, and it's really good for looking at MRD and therapy response. SAGA Seq is our NGS-based solution when you want to look at hundreds of targets at a time. But the main subject today is SAGA Safe, which is our patented chemistry for digital PCR that will improve standard digital PCR by about 100-fold. And this is ideal for looking at things like companion diagnostics and monitoring specific mutations. Our customers today are mostly pharma companies as well as some hospitals and clinic labs. We have over 250 assays, and we CE marked our first one last year for the EGFR T790M mutation that Evie mentioned. SAGA Safe is compatible with all of the digital PCR machines we've tested so far, Bio-Rad QIAGEN, the Combinati Thermo Fisher box, and of course, now with Stilla. And what we are seeing is that when you want to have very high sensitivity in a complex multiplex assay, SAGA Safe is really a terrific technology to allow you to have complex assays, but have them also retain high sensitivity. So the way SAGA Safe works is it basically achieves better performance by minimizing the background noise. That's shown here by these red dots indicating that you can get false positive mutations using other methods like NGS or even standard digital PCR. And that false positive noise translates to worse performance in terms of lower limits of detection. So shown here in this dilution series of expected MAF and measured MAF, when you get to very low expected MAFs, you start to run into the noise of competing methods. And that's where the SAGA Safe technology allows you to really measure things at these very low MAFs down to around 0.001%. And as I mentioned before, we've shown that this really works and improves any compartmentalized digital PCR system. So where is this noise coming from? There's 2 major sources. One is from the polymerase enzyme itself. When it copies DNA, it makes errors, and that affects NGS and Qpcr and digital PCR. And there's also DNA damage can also cause noise, of course. The way SAGA Safe works, just to in a limited amount of time, is that we are able to, within one reaction, all in one, you put all the components in at the same time, but you can actually run the test or the assay in 2 phases, where in the phase 1, you have a linear copying of your target strand. So you maintain sort of perfect copies where the DNA polymerase error basically doesn't happen, or if it happens, it just happens in 1 copy out of many dozens of copies. And then in phase 2, you then take those resulting linear copies and you generate the signal to see what was the true state of the starting molecule, target molecule in that compartment. And what this leads to is basically you can reduce your false positives by about 100-fold, which means you can increase your or improve your limit of detection by about 100-fold. And another benefit is that you can also quantify single-strand DNA from double-strand DNA. So here's some competitor data showing that when you look at pure wild-type DNA, you have these false positives, whereas with SAGA-safe assay, the exact same input DNA, it's very clean like this, and you don't have any false positive signals. And I'll just point out that this down here, some people say that this looks like you have heavy rain, which is a common digital PCR phenomenon, this idea of rain. But actually, this is not rain. This is a positive side effect of our technology, which measures the single-stranded target DNA. I don't have enough time to go into what this means exactly, but it's actually quite beneficial in terms of comparing data between platforms, for example. So with false positives, you have a limited detection of around 0.1, but with SAGA-safe, you can really push this down to the 0.001% range. And this is in the a single-plex format. So when you have a high sensitivity test, it means you can find more patients having mutations. So with NGS, across 200 breast cancer patients, looking at 3 PIK3CA hotspots, we found about a third of patients had a mutation. Whereas with SAGA-safe, we found over half of the patients had a mutation. So with more sensitivity, you of course find more patients, and we've done biological correlates, and they're very significantly associated to ER and PR positivity, which shows us that this is not noise. It's true mutational burden associated to known biological parameters. So in this project, our objective was to adapt a SAGA-safe PIK3CA assay to the Stilla Prism 6-color system using their Sapphire chips. And just to jump to the result, we will, as you'll see, show that this proof of concept 24-plex PIK3CA assay works really well and has really great performance and very nice LOBs and LODs. And so in conclusion, we know now that SAGA-safe is fully compatible with Stilla, and that having the ability to use 6 different color channels is great. But to really achieve high sensitivity in a complex assay, it really helps to have an ultra-sensitive chemistry to begin with. So these are the 24 different mutations that we looked at. I won't go through all of them, but basically these are all the relevant PIK3CA mutations. The 3 most common are the ones I've indicated there in bold. And you can see on the far right which channels we are placing the different signals into. So here's some data looking at all 6 color channels. So on the x-axis on all of these plots is the red channel, which is looking at wild-type DNA, and the y-axis are the 5 other different channels. So this is looking at pure wild-type DNA. So we expect to see no mutations, and that's basically what you see here. That's very clean data, and it's very accurate in quantification. Here's a couple examples of dilution series, a serial 1/2-fold dilution series. You can see the R-squared values are extremely high, showing that this is very quantitative down to around the 0.01% range and lower. We looked at a number of samples where we had NGS data and ran some breast cancers through this with the Stilla assay. And you can see very good concordance between the NGS maps and the Stilla Prism maps. And here are some examples of 3 mutations from this plot here, showing what the data looks like. And it's very straightforward in terms of thresholding and so forth. I think this is one of the most, my favorite slides here, because it really shows that you can achieve terrific performance in this kind of complex situation across many color channels. So LOBs are in the 0.003 or below range, and the LODs are just under 0.01%. And here's an example of one mutation, for example, where you can see good detection of this Q546P mutation at a MAF of 0.009. So we had this objective, and we actually only had about 3 months with the box. So this was done rather rapidly. We're very happy with these results and the quality of the data and the performance of the new 6-color Prism machine. Yeah, we're looking forward to continuing to use this and to generate more assays going forward. Thank you very much.
Thank you, Lau. That was very, very interesting as well. It's amazing to see that you could develop an assay targeting 24 mutations in 3 months, and that it works so well. I mean, it tells a lot about the quality of your chemistry as well. So great combination there. Maybe I can ask the other speakers also to come back on the stage. While we do that, I'll just start by asking you one question, Lau, which is, as we see the increase in the multiplexing capabilities of digital PCR, how do you see the interplay between NGS and digital PCR moving forward? And do you see a limit also to the level of plex that you can achieve in digital PCR?
Okay, I'll take the first part of your question, which actually has a kind of a complicated answer. So I think there's certainly going to be a lot of interplay between NGS and digital PCR. I mean, there's lots of room for both to coexist. And I think there are use cases in cancer where you may do an upfront NGS to sort of to do a wide screening of all the expected variants. And then in follow-up testing, maybe switch over to digital PCR once you know sort of what's relevant for that patient. That's, for example, one approach. We have another approach that also marries NGS and digital PCR, where we use NGS to find chromosomal rearrangements, which are actually, we think, better markers of MRD than point mutations. And then we use digital PCR for chromosomal rearrangements to monitor MRD in a quantitative way. And then there are instances where, it'll be NGS all the way. And I think there'll be some instances where you don't need NGS at all, and it'll just be a pure digital solution. So it depends a little bit on the cancer form and the clinical question at hand.
Okay, thanks a lot. I was going to ask a similar question to the other guest speakers, but you had one question directly targeted at you, Lau, and from the audience, which is, are all the kits from the SAGA Safe range compatible with Prism 6, or just the PIK3CA that you showed?
I think in principle, they all can be compatible. We have not run them all on the Stilla box yet. We've focused only on the PIK3CA thus far, but it transferred over to the Stilla machine very quickly for us. I think partially because we have got great people in the lab, but also because the Stilla box is pretty easy to work with. So we think that all of our assays, could work on the Stilla box with sort of minimal optimization from our end.
Okay, great. Thanks a lot. So maybe we'll take 10 more minutes to do a round table. So thanks, Herman and Evie, for being back on the stage here. So I maybe want to start by asking the same question I asked Lau to you two, which is, how do you see the interplay with NGS and between NGS and digital PCR as we expand the multiplexing capabilities of digital PCR? Who wants to start? Herman, maybe?
Yeah, I can start. Yeah, I think it goes beyond confirmation. Like some people use the Crystal droplet PCR to confirm next generation sequencing. In our case, we are using we are trying to use like Crystal droplet PCR for a specific assay. In our case, to measure at genomic copies of HIV. That is a very complex system, but we can do very high throughput, so we can see the structure of HIV reservoir. So we are talking about genomic DNA copies of different genes in one droplet, right? So we will know if the HIV reservoir is intact or defective. And then we can use next generation sequencing, for instance, to look at immunological signatures or different interferon pathways, whatever biological processes in gene expression assays. So it's not going to be like technical to confirm what we saw by next generation sequencing. It's going to get biological processes. Next generation sequencing, knowing the immune signature of gastrointestinal tissues, lymph nodes, and crystal droplet PCR just to look at the structure of the HIV reservoir. This is one application that I think.
Right, thanks. Evie?
Yes. Personally, I think in my field, in liquid biopsy and in cancer, as Lao very clearly pointed out, and it's correct, it has to do with what is the question. And if we start with some patients, it's good to have NGS in the beginning, just to see what are the main alterations that we can follow up in each individual patient. But then it's better to have a very specific assay that it's highly focused on specific alterations that we need to follow up for each patient. For, let's say, for efficacy of drug specific treatment, for resistance mechanism like PI3 kinase, as correctly pointed out, or ESR1 mutations, or EGFR mutations, then you definitely need a PCR-based based approach, because it's highly standardized, much easier. You can have an end user in each lab. And I strongly believe that the main ultimate, let's say, goal of liquid biopsy efforts is to bring liquid biopsy in clinical practice, like in clinical chemistry, where we need to have the Levy-Jennings graphs, we need to have standardization, we need to run this in a routine base. And this cannot be done with NGS. Yeah. So I think that it depends on what we want to do. Mm-hmm. And NGS provides a lot of information. It's highly important. But to end up in the clinical setting, in the routine clinical setting, we need assays, multiplex assays for sure, that we need to see a lot of markers. Not only mutations, epigenetic levels, methylation as well. We have to do a lot of things, but to focus on the panels that we need to get information.
Yeah, and I think that makes a lot of sense, and the ease of use and the deployability of digital PCR, Crystal Digital PCR, makes it very well suited for clinical applications. I'll turn to another questions we have. So people in the audience who are interested in understanding how the Stilla system compares to other digital PCR solutions beyond just more multiplexing. So I'll maybe rephrase a bit the question, and you've all used the system, so maybe you can comment about the usability. And also, for those of you who have designed assays, if you can share the assay design experience and how that went, as obviously this is a key step in bringing any new test to samples in the clinics. Who wants to start? Lau, you were the last to speak.
Yeah, sure. So we've used pretty much all the instruments in our own hands, and I mean we like the Stilla box a lot. It's a little bit difficult to compare and contrast, them all, but I mean Stilla with 6 colors with the prism. I mean that's as good as it gets. That's the most color channels that's available right now, and I think the software works really well, and because when you look at 6 dimensions, it's actually it opens up a whole new way of thinking of this data, which we've just started thinking about in the last couple of months, and it's actually quite interesting. So you of course need to have good software for that. In terms of assay design, I mean we've been doing this for like 10 years. So I think for us, we can do things quite quickly and well. And SAGA safe design is actually quite different from your standard TACMAN style design. So we have our own way of designing things to achieve the kind of results that we achieve. But we were actually very happy about how quickly our existing designs sort of worked with very little tweaking, transferring them over to the Stilla machine.
Mm-hmm. And you have maybe experience in converting Qpcr assays to digital PCR. To those maybe in the audience who have that in mind, in your experience, is it easy to do, and is digital PCR maybe a more lends itself better to the designing multiplex assay than Qpcr?
Is that a question to me?
Yes. Just
yeah. Actually, we kind of hopped over Qpcr to some extent. We don't have a whole lot of experience to compare, but in general, I mean, when we were looking for a quantitative method and chose to sort of start using digital PCR many years ago, one of the benefits of it was that the quantification not requiring reference standards, that you get direct copies readout, which I think Evie, you pointed out that to compare between different hospitals and labs, it's actually a little bit hard to use math. I mean, NGS can only give math generally to give copies. With NGS, it's not very easy, but with digital PCR, you can get an absolute quantification. That's I think that'd be very useful to look at not just changes in math, but changes in the absolute number of copies. I think we're seeing early data saying that actually copies is perhaps having a little more information than just math.
Okay. Herman, maybe you want to share your experience, and you also have experience with Qpcr. I mean, you have
yes. I mean, initially,
yes.
Initially we developed assays like to measure HIV reservoir and to be honest, we use Biorad, the competition, right? So we published a paper this year in Cell Reports Medicine, our lab, on it's a dPCR assay just to quantify the structure and numbers, and that was pretty good. So we published this year, and now we purchased the 6 colors. So we're trying to adjust the multiplexing. We've been talking to, I forgot the name, Gutierrez in the company,
yes.
Back and forth. Kim, maybe she came back and forth. So we're trying to now use that to multiplexing. This is one side of the story. All the work that I did with opioids, I also did in the beginning. I did it part of them with. I did it in Biorad, and then I compared the data with naica. It was identical, so it really worked very well. But I prefer your software better. And in terms of real time, I developed the assays initially in real time because it was like very fast and cheap. So it took like half an hour if I do the fast master mix. So I got good data in the real time, and then I compare with the naica, and it was excellent correlation between CTs and copy numbers. The only challenge that I have with digital droplet PCR is with the normalizer that I use in these Siemens samples, because sometimes I couldn't do duplex, let's say one of these tRNAs and normalizer in the same sample, because the normalizer will saturate the signal and the TRF no. So I had to tweak a little bit the experimental design, but I made it to work. Yeah.
Okay. So if I understand correctly, you have a simple workflow, which is doing the quick and dirty way of Qpcr to design, and then you switch to dPCR quickly, and you get the value of sensitivity, precision.
Exactly.
Really
And
nice.
it correlated very well. The CTs, I didn't show in the presentation, but it correlated very well, the CT with absolute copy number. It was perfect.
Okay, very interesting. Evie, maybe to conclude.
Yes, from my point of view, yes, we have many years experience in designing assays in Qpcr, and our experience has proved very valuable in bringing these assays into digital, which was easy for us. And I would say that it's easy to bring the primers and the testing and everything from RT-qPCR or from Qpcr to digital. We have publication this year using dPCR, RT-dPCR for PDL1 in clinical chemistry, where we developed an assay that we actually transformed from RT-qPCR to digital. And it worked pretty well. And moreover, the good thing with multiplexing is that with digital PCR and Crystal Digital PCR is multiplexing, and the use of these 6 channels enables to include one external standard in all these procedure that is highly important, as I showed in the Levy-Jennings graph. And of course, with multiplexing, you can save sample. So I think it's pretty easy to go from Qpcr for the users that have Qpcr in their labs. And my lab is for more than 20 years on Qpcr, and we have transformed the last 5 years to digital. And it's pretty easy, and absolute quantification is an important issue because we don't need the standards. We just have absolute quantification. So I see a lot of potential in diagnostics in this technology, high potential.
All right. So thanks, everyone. I think we're going to wrap it up here. I think we were aiming for an hour, and I think we're already 10 minutes over time. I think it's been really interesting hearing all of your applications and uses of the naica system, pushing the limits on the high-plexing in digital PCR. And thank you again. Thanks to our audience for listening in. And if you have questions that haven't been answered, don't hesitate to ask them by email. I put my email address on the chat box, and we'll do our best to answer those offline. Thanks again. I look forward to seeing the fantastic research results that will come out and the products, clinical products as well, we call them out of your labs. So thanks, everyone. Goodbye. And have a good day.
Thank you very much.
Bye.
Pleasure.
Thank you.